Using electroencephalography, or EEG, the measurement of electrical activity on the scalp, behavioral neuroscientists try and understand and measure basic functions of the human brain. Now, doing some of these advanced procedures, we run into some significant computational bottlenecks, and this brief talk is just about one of these measurements that we're trying to overcome and establishing good compute resources for electrophysiology. So here's a basic problem that we have. We have a model of the human brain here, the cortex inside of the skull, and then inside the skin, and we can basically look at different parts of it. These dots represent areas where the brain is becoming active. And what we want to understand is, in a very small timeline, within a second of a certain stimulus, how do these different regions become active differentially across groups or across conditions, and how do they influence each other? And what we can do is watch patterns of the interaction between brain regions over a very brief period of time with a very high resolution. Now, doing this sort of analysis with EEG, there are major hurdles to pass in order to get this sort of spatial resolution that we want to have and look at interactions between regions. The way that we do this is a form of unmixing. It seems to be one of the best ways of doing this. Is called the independent components analysis. And what I'm going to do is give you a demonstration of this because it's very computationally demanding, and this is one of the main problems that we have in terms of being able to do research in a realistic amount of time. So imagine you have a room, and around the room you have a whole bunch of microphones. In our case, the room is a human head, and the microphones are actually sensors that are picking up electrical activity, but it's the same thing as picking up sound around a room. These green dots represent the location of just over 110 sensors on a human head, and this shows where they are in space. Another view of this is looking at it straight from the top, where this is the nose, the left ear, and the right ear, and these are all of the sites. Now imagine we were trying to record musicians inside this room. We don't know exactly where they are, but there's several musicians in there making sound, and we're going to pick them up. At the same time, though, there's somebody hammering on the wall in one location. There's a bunch of people talking. And what we really want to do is get the sound from each musical instrument alone. So right now, while we're recording sounds, at each location we're picking up the sound from every source of sound. What we would actually like to do is plug our recording device right into each musical instrument and just pick up the sound of that one thing. Now, independent components analysis is able to replicate that in some ways. So really what we want to do is, rather than picking up the electricity on the scalp and look at a signal like this, each one of these lines corresponds to one waveform on the scalp. And if we look close, we can see that there are these patterns that go across multiple sites. These big blips that we see here are actually eye blinks. This is kind of like a person hammering on the wall inside of a room. Now, if we look at how these big blips look across sites, we get something like this. We get a big voltage right over the eyes, the front of the face. Now, if we take these signals, what we really want to do is we want to isolate this pattern that's going across the front of the head. Remove that because we aren't interested in it. So we do independent components analysis. What independent components analysis does is, instead of returning a site of a location where we're recording all of the sound from, it isolates a particular pattern. It says, look, there's this pattern. I'm going to isolate the signal coming from that. And then what we get is this, where we see that the blink activation, these big spikes, aren't going across signals anymore. It's completely isolated to this one waveform. And what we can do is we can just decide to remove it. Thus, in our recording of the room, if this were the sound example, we would be getting rid of the person hammering on the wall. And then what we do is we actually maintain the activation of the others, actually isolating sources of information in the brain, like plugging in directly to each musician. And here we have an example of the locations of each source of information that seems to be coming from the cortex during this specific task, as though now the recording sensors were located right inside of the head in important regions of the cortex. Importantly, these are also not picking up everything around them, but directly picking up only what's there, as though not only is the location of the recording more close, but it's only picking up that source, as though we're plugging directly into the musical instrument in our example. And then once we have these sources and they're independent, that's when we can get back to the real goal of looking at how brain regions interact with each other at a very high time resolution.